So I'm going to call Susan, and somebody said you were 3 tables over. I don't know where you are, but the chair of the board to come up that way, okay? So before we get on with the research presentation this afternoon, we've got a couple of awards to give out. This is the National Award of Merit, the corporate award, and 2 are being given out today. Our first one is being given out to R.A. Utting and Associates. R.A. Utting and Associates has truly demonstrated a willingness to help in many ways, as many of our donors do. Please help me congratulate R.A. Utting and Associates. Susan Van Arsen will accept the award on behalf of the London chapter and present it to R.A. Utting when they get a chance. A little bit of information before I call Susan up. For more than 12 years, R.A. Utting and Associates have been involved at the national level, creating a family fund and contributing locally, also through the London Indie, various barbecues, bowling events. When the London chapter applied for Stay Strong funds for recent events, it was the Utting family that reimbursed the fund for those expenses. R.A. Utting and Associates sponsored the last 3 London golf tournaments. Thank you very much to R.A. Utting. We could not do what we do without the support of our corporate sponsors. Thank you so much. The second recipient has truly demonstrated a willingness to assist in multiple ways as well. Selkirk Record. Please help me congratulate the Selkirk Record this evening. Jeff Bose of the Winnipeg chapter will accept the award. Jeff will take this award home and present it to Brett Mitchell for us. To welcome our next presentation, Dr. Ray Truant and Dr. Tamara Maiuri. Both have been incredibly involved with the Huntington Society of Canada and with the research we've done over the years. Dr. Ray Truant has been with McMaster University since 2000. He has been a recipient of the CIHR New Investigator Award 2001 to 2006 and a fellow of the Howard Hughes Medical Research Institute, Duke University, 1996-1999. Dr. Truant received his PhD in Toronto at the C.H. Best Institute of Biomedical Research and is supported by CIHR. His career to date includes over 50 career manuscripts. Dr. Truant was a recipient of the Queen Elizabeth II Diamond Jubilee Medal for Public Service in 2012 and is now a full professor at McMaster University and chair of the Scientific Advisory Council for HSC and has been for many years. As well, he is an external scientific advisor for HD Buzz. Ray has been incredibly instrumental in helping us raise research dollars for Huntington Society as well. He's always open to me taking donors to his lab so that they can see how it's done. You might be surprised that people don't work as much at the bench anymore. When I go to Ray's lab, we're going in dark computer rooms where his master's students and postdoc students are sitting in dark computer rooms all day. Wow. It's much different looking now, but Ray has been incredibly instrumental in helping us make research much more tangible for everybody. Dr. Tamara Maiuri is a research scientist in Ray's lab. However, she's much more than that. I'll get through the technical stuff here and then talk about how much more she is. Prior to joining the Huntington's disease field, Tamara obtained her PhD from the Medical Biophysics Department at the University of Toronto, where she studied the cell biology of cancer-related genes. Her work in the Truant lab focuses on the normal biological functions of Huntington protein in hopes of understanding how they may be disrupted upon inheritance of the mutant Huntington gene that causes HD, and how they may be restored by small molecule drug candidates. Tam is also in a band, Eli and the Strawman. And about 2 years ago, Tam sent me a note and said, you know, I'm in this band and we would like to raise awareness across Canada for Huntington disease. And we'd like to create a song, we'd like to go across the country, would you be interested? And after the fact, I understand that Tam's father said, don't ask, they're not gonna go for that. And I was really excited about it. I thought, oh my gosh, having this talent with this expertise, the whole mesh. So Tam, they did a road trip in the summer and went from coast to coast, increasing awareness about Huntington disease. Magnificent, and they were part of our PSA campaign. In her spare time, when she's not in research or doing the band, Tam also writes and reviews peer research for HD Buzz. So, as does Ray. It's incredible. Anyway, I would like to invite both Tam and Ray up, and the title of The Start of a New Era in HD Research.
Okay, so we're going to do this as a tag team show. I'm going to give you a little bit of an intro and a bit of theory as to what's been going on in Huntington's disease research, and then am I getting this? And then I'm going to hand it over to Tamara, because she's the person who's been doing the real work on the bench and actually driving a lot of the intellectual questions that we've been asking in the lab for the last 2 years. We're really excited to give this talk. Things have, in my opinion, we've undergone quite a transformation of understanding of Huntington's disease in the last few years. And we've got some fundamentally new pathways to look at that we were not talking about 5 years ago. And hopefully I'm going to show you some exciting data that will tell you that there could be a benefit to this sooner than later for HD. Okay, so much like Jeff, I'm going to start with a historical photo here. And many of you know who this is. Put up your hands if you know who these guys are. Oh, come on. So that's Watson and Crick, right? So these are the guys who won the Nobel Prize for the discovery of DNA, the structure of DNA. My thesis supervisor at University of Toronto was Jack Greenblatt. His thesis supervisor at Harvard was Jim Watson, right? And when I was a grad student, I had a nice dinner one time in Toronto with Jim, and I asked him, I said, well, you know, if you had to be a grad student now and start your career in science, what would you work on? And he says, well, if I were you, I'd work on brains. So I kind of heeded his advice. Okay, so there are solutions that we're looking at for Huntington's disease, right? And it's great that Jeff gave that talk, and I hope a lot of you, or if not most of you, saw it, because he covered a lot of the background that I'm not going to have time to cover. But there's some very simple concepts that we have in terms of drugs, right? So we know production of the mutant protein is bad. It's what causes the disease. Unlike Alzheimer's and Parkinson's, we know this is absolutely true for Huntington's disease. So let's just get rid of it. Let's silence the gene. That's one approach, and Jeff talked a bit about that. There's been other approaches chemically with companies like Eli Lilly and Pfizer, where they know that there's been some hyperactivity of some pathways within the brain And if we could inhibit those may be beneficial in HD. And those have gone through clinical trials. And we're just waiting to hear the open results for that. In my lab's situation, we're interested in small chemical groups that should be on the mutant Huntington protein, but are not. And then we're trying to see if there's chemicals that can allow those chemical modifications to be restored to normal. And lately, what this work has led to is a concept that maybe with these small molecules, we can actually fix the mutant protein, turn it back into the normal function of the normal Huntington protein, which is very different than silencing it. Okay, so the hypothesis that's been going around for the majority of these neurodegenerative diseases, this isn't just Huntington's disease, but also Alzheimer's and Parkinson's disease, and versions of these diseases, frontotemporal dementia, etc., is the amyloid hypothesis. The idea, and this has been about 20 years old, 25 years old, to the forefront, that we have the situation that proteins are being misfolded, right? So a great example of protein folding is when you cook an egg. Crack open the egg white is nice and clear, and it sits in the pan, and then it goes opaque. What you're doing is misfolding. You're refolding that protein from clear, and then when it goes opaque, it's in a completely different folded form. It's actually precipitated out, and that's why you can't see through it. And this has been the leading hypothesis in neurodegeneration research, right? This idea that if we look in the brains of people who have died from these diseases, we can see all kinds of evidence of accumulations of misfolded proteins at very high levels. In Alzheimer's disease, it's this beta amyloid protein. In Parkinson's disease, it's a protein called alpha-synuclein. And similarly, in Huntington's disease, there's been a lot of people thinking that the problem is the misfolding of the mutant Huntington protein, right? And so we've been now working around these hypotheses for the better part of 2 to 3 decades. And we're starting to realize there's problems, right? So the problem, especially from guys like me, I'm a classically trained biochemist. I'm not a neuroscientist, right? In 25 years, no one has ever come up with a really good explanation of how these misfolded proteins persist and hang around so long in the brain. How is it that it can take 40 years to get sick? Why, if it's a protein misfolding problem, isn't it happening from birth? And why are people getting Alzheimer's disease at age 10 and 12 instead of 40 to 65? So there's actually never been really strong evidence that says that those spots that we see in the brains cause the disease. We know they're in diseased brains, absolutely true, but is that just a correlation, or is one really causing the other? And there's evidence on both sides of that to suggest it. So it's controversial, right? We can't say it's right, we can't say it's wrong. But especially in beta amyloid, as people look harder and harder in brains by new methods and younger people, we're getting big surprises. So when we start looking at sports players who take a lot of head hits, like hockey players, these guys, right? What you're going to see is that their brains have a lot of beta amyloid deposition in them. It looks like, if you were just looking at amyloid, these guys look like they should have Alzheimer's disease, but they don't. And similarly, you can find elderly people that have a very high load of some of these plaques in their brains, but don't actually have dementia. And that's traced over to drug studies where several groups have now done clinical trials where they've met the burden. They've come up with a compound that we can give to a person that will lower the number of plaques that we see in the brain by up to 30%. But there isn't an obvious clinical output. And those are still being trialed now, and that may just need some tweaking, but it hasn't given the obvious answer that we thought it would, right? And this has actually led to certain people, and I'm quoting here the late Dr. Mark Smith, who's a friend of mine. He was the editor of the Journal of Huntington's Disease, and one of the last articles that he ever wrote prior to his death was this concept that the amyloid hypothesis might be too big to fail. We're heavily invested in one idea. Maybe we need to look at more alternative hypotheses. And in fact, now, in the press, the word hypothesis has suddenly reappeared behind amyloid, whereas for a long time, this was taken as fact and that it's the cause of disease. Okay. The other difference is we base a lot of our research on mouse models, right? Mice are not people. So mice, they live 9 months in the wild on average before something eats them or they die. They don't age like humans at all. And we know that historically, in mouse models where we can get drugs that actually fix the mouse, the actual success rate in humans is actually quite low, right? But it's the best we can do in terms of mammalian models. So for example, in the HD mouse, we need to put in very long CAG alleles, over 100 repeats. And that is actually rarely seen in human beings, right? We give it a really extreme version of the disease in order to model it in the animal. And then when we do this, we do get an animal that looks like it has various aspects of Huntington's disease. It doesn't walk right, and a whole series of changes that look like HD, right? In humans, though, we get old. And when we get old, our energy centers in our cells are slowly burning out. Our batteries are running low. We're just not able to produce the level of energy that we could when we were in our earlier years. And we have a real problem in humans that as we get up above the age of 40, we're getting an increasing buildup of toxins that are coming out of the process that are not getting cleaned up as they are when we're younger. And so in the clinic, in real HD, the typical allele is not over 100 that we have to use in the mice. It's only 42. But if we take those 42 and stick it into a mouse, it's very difficult to see disease. We can do it now, but it's not that obvious. And we get things that happen in humans that we don't really see in mice. We get wrinkles, we get gray hair, we get loss of muscle, we get loss of hair. I'm not in denial, I know this will happen to me one day. I will turn gray. There will be some hair loss. And part of the problem with all of this is new to medicine. Before 1920, the average age of death was about 42 to 45 years of age. Nowadays, your kids, especially that noisy one in the back, that kid's going to live to 100 years of age on average, probably, we predict. And this is a whole new era of medicine that we just don't have experience with. People didn't live this long. And a lot of that is due to medical advances, right? So we have to understand it a bit better. And this is where Tamara came in, right? And one of the first observations that she realized was that our models that we're using in this disease are disconnected from what's really going on in the clinic, right? And so here's a typical graph that so many of you have seen this before, right? And Jeff pointed this out. More CAGs is better, right? The longer the CAGs, the earlier you get to the disease. But there's something very interesting in humans. Humans are not all the same. We're not mice. We're not on a genetically similar background or genetically identical background. If you look at what happens at the typical clinical alleles down here around 41, 42 repeats, there can be as much as 3 decades of difference when the age of onset of the disease. And similarly, when you look at the profile of the alleles that appear in the disease, you can see that most of them are hovering around this 42 to 45 range, and that these extreme alleles are actually extremely rare and are not age-onset disease. These is juvenile HD, where kids get sick from a very young age. So, back to the geneticists. These were the heroes of 1993 that gave us the Huntington's gene and gave us so much hope and started the research on real molecular mechanisms of this disease. They went back to the Venezuelan cohort, and they went back to additional numbers, 4,000 HD patients. And we wanted to ask the simple question, what is it about the other genes in these people that can make the disease so different between one person and another, even though they have exactly the same number of CAGs? And this is something called a Genome-Wide Association Study, GWAS for short. And they released this data and they published it in 2015. And we got a list of pathways that really influence the course of disease in the clinic. This isn't models, this is real people, right? And there was a big surprise. This is not what we expected. We expected to see a bunch of stuff related to the amyloid hypothesis. What we got, which led to very confused generic HD researchers like this stock photo here, is pathways that were involved in mitochondrial function. So that's the energy center that I was telling you burns out as we get older. It may cause gray hair in some of us. DNA repair turned out to be by far the most common pathways affected, almost all of them. And what's left are a few interesting pathways that are revolved around something called redox, which is these toxic products that occur as a result of oxidation of DNA. And what we didn't see was anything related to amyloid. And this has led to a lot of confusion, where people are sitting back saying, wait a minute, what really is affecting the course of the disease in humans may not be exactly what we thought it was. Right? Okay, so this is where Tamara's going to step in and tell you what she's been working on.
Okay, so Ray's going to come and talk to you a little bit more after I explain all about this idea of oxidation and DNA damage. And he's going to talk about some of the most exciting stuff happening in the lab. But first, I'm going to delve a little deeper into this idea of oxidation. And as Ray explained, as we age, this natural oxidation is kind of like rusting. You can think of rusting. Rusting is an oxidation reaction. And so it just happens naturally. And it will damage the gears that make our brains work. So these naturally occurring oxidants that accumulate as we age, they'll damage proteins, they'll damage fats or lipids in the brain, and they'll also damage DNA. And that's critical because the DNA acts as a blueprint for all of the stuff that our cells need to make. So if we just go back for a second, these three groups of genes, mitochondrial function, DNA repair, and redox control, three groups of genes that were the most common genes that came out of the GWAS study, which was asking what are the other things that affect whether somebody gets sick early or late. These three groups really make a trifecta of oxidation and fixing oxidation, because the mitochondria are the primary source of oxidants in the cell, especially if they're not working properly. The redox control genes are in charge of sort of mitigating damage caused by these natural oxidants. And DNA repair genes fix the damage. And because it's so very important to maintain undamaged DNA, our cells have lots of ways of doing this. And we're constantly repairing DNA, and we have a whole host of genes dedicated to this task. And when things start to go wrong with those genes, that's when we get disease. So DNA damage in cycling cells can lead to uncontrolled cell growth, and that leads to cancer. DNA damage in non-dividing cells like neurons, the opposite happens, and the cells die. And so this buildup of DNA damage is a problem in other neurodegenerative diseases too, like Alzheimer's and Parkinson's. And so we have this host of genes dedicated to managing oxidation and repairing our DNA. And so you can imagine that if some people might have some genes that are slightly better at their job, and other people might have genes that are slightly worse at their job. And that's what was reflected in that GWAS study. So one way of interpreting the GWAS results is that maybe these little differences in genes that make the gene slightly better at its job or slightly worse at its job might be slightly protective, or might make cells die a little bit earlier in those very vulnerable neurons in HD patients, because they're already on the verge because of toxicity from the HD gene. And that would be a very reasonable way to interpret those results. But what I'm going to talk to you about results from our lab suggests that there's a little bit more to the story, because we've found that the Huntington gene is actually directly involved in DNA repair. So I'm going to tell you actually about 4 different projects. Well, I'll tell you about 2 of them in detail, and then I'll gloss over the rest. But there's 4 different projects that seem to be unrelated at the outset. But once we learned from the GWAS study in humans what was important to disease onset, DNA repair, and oxidation, these 4 projects kind of converged on this topic. And I'll tell you about that. But before I do, I'm going to do a little review of the HD gene, the CAG repeats, and how that translates to the HD protein. Because that's really what we work on in our lab, and I know for those of you who came to Ed and Jeff's talks, we talked about it already, about the gene and the protein, but so for those of you who already are familiar with how genes control, what comes out in our bodies, this will be a review. For the rest of you, consider it a crash course, and at least at the end of it, I hope that you'll understand why the number of CAG repeats is so important to us. Okay, so let's get down to basic bio. So every cell in our brains and almost every cell in our bodies has 2 copies of each chromosome. What a chromosome is simply a long string of DNA. And genes are regions on that stretch of DNA. So we inherit 2 chromosomes, we inherit 2 copies of each chromosome, one set from mom, one set from dad. And so what that means is we inherit 2 copies of every gene, one from mom and one from dad. And that includes the Huntington gene. And each copy that we carry has a certain number of CAG repeats. Okay, so what are the CAGs? Well, the CAGs refer to the building blocks that make up the string of DNA. They're strung together like beads on a string. There's actually 4, T, C, A, and G, and they're all in different orders. But in the Huntington gene, there's a part that goes CAG, CAG, CAG. And so if both of our copies of the Huntington gene have 17 repeats, no HD. But if one of our copies has more than 37, we will get HD at some point in our lives. And that depends not only on the number, like Ray explained, but also on this host of other genes that we inherit that are involved in oxidation management, DNA repair. So how does the CAG then cause HD, Huntington's disease? Well, we don't know the exact answer to that yet, but to think about it, we need to think about protein. And so that string of DNA is translated into protein. And there's a code. For every 3 building blocks in the DNA, you get 1 amino acid in the amino acid chain. And that chain folds up into a protein of a certain shape. And the order matters because the shape it folds into depends on the order of the amino acids in the chain. And so we have these proteins folding up into different shapes to do their jobs, thousands of them in the cell. And they all fold up differently. One protein might fold into a long structural rod, and its job is to add strength to the cell. A DNA repair protein, for example, might fold into something that acts like molecular scissors and snips out the damaged part of the DNA. So the shape is important because if it's not the right shape, it can't do its job. And the protein it doesn't stick around. It's constantly being degraded and turned over. And fresh protein gets translated off of that DNA template. So you can see why it's so important to protect the DNA, because it acts as the blueprint for everything that our cells need to make. And the code that we inherit from our parents dictates the sequence of the amino acids, which dictates the shape of the protein, which dictates how well it does its job. So you can probably guess where we're going with this. What happens when you have too many CAGs in that gene? Well, what happens is you get extra amino acids, and that's going to change the shape of the protein and make it harder for it to do its job. Okay, so what is the shape and the job of the Huntington protein? So the job of the Huntington protein is to act as a scaffold. It's not really that exciting. It actually just hangs around, and lots of other proteins will come and interact with it. But its job is really to be there and make sure that other proteins can be in the right place at the right time. So much like a scaffold will allow workers to come together and pass tools to each other and have access to whatever it is they're working on. That's the Huntington protein's job. And both Ed and Jeff had a picture of the actual structure of the Huntington protein, which I didn't have time to put in here, but some of you may have already seen it. Kind of looks like a blob, but it's a hollowed out blob. And so it's not, if we're thinking in terms of a scaffold, you can kind of think of it as this hollowed out blob, but it has regions within it that are flexible. So it can take on a lot of different shapes. And that makes it a very versatile scaffold. So the cell uses it for a lot of different jobs. Sometimes it might use it to bring proteins together at one place and one time to do one job, and then another time, totally different proteins in a different place in the cell at a different time. And that versatility makes it really hard to figure out what's going wrong when you have extra amino acids because of those extra CAG repeats. Because the Huntington protein has so many jobs in the cell. Some of those jobs might be working just fine. And so that basically is getting to the root of the problem. Which one of Huntington's functions is important in HD? And then the next obvious question is, how do we fix that problem? And we have a few approaches to this in the Truant lab. So one approach is to look at normal Huntington, see what it does in the cell, figure out what its jobs are, and then ask what happens when you have those extra amino acids because of the extra CAGs. And another approach is to kind of skip that step altogether and just screen drugs and look for things that affect the Huntington protein. And we don't do it like totally blind. We have a few things that we look for, like where the Huntington protein goes in the cell, or maybe what kind of chemical tags get added onto it. But those are our two approaches. And so now that we've covered the gene to protein to we study the protein idea, now I will tell you about what we're working on in the lab. So these 4 seemingly unrelated projects that were all underway before that GWAS study was published. And so once the GWAS study was published and showed us the importance of DNA repair genes and genes managing oxidation, at that point, we kind of knew what was going on with these other 4 projects. But basically, Laura Di Giovanni, a recent grad from the lab, she was asking what happens to Huntington when you throw oxidants on cells. I was actually following up on a publication that showed that a DNA repair protein was involved in HD. Susie was following up on a previous student's work that was trying to understand what controls the Huntington protein, where it goes in the cell. And Laura Bowie is screening drugs for things that affect the Huntington protein. So, like I said, this was in motion before the GWAS results told us that oxidation was something that affects, when people get sick. But one thing that we did know was that in response to just about any stress to cells, the Huntington protein moves to the nucleus. So you can heat the cells, put them in ice, shake the plate that they grow on, whatever it is you do to stress the cells, the Huntington protein goes to the nucleus. But when you think about that in terms of disease, probably the most important stress when you're talking about aging neurons is oxidants. This is known. So Laura's job was to look and see what happens to the Huntington protein when you add oxidants. And what happens is, same thing that happens with other stresses. The Huntington protein moves to the nucleus. So what you're looking at here is actually real Huntington protein in cells that were isolated, skin cells that were isolated from a patient or volunteer from the clinic. So these are not mouse cells, and they come from real people. And this is the actual natural Huntington protein that lives in the cell. And the way we see that Huntington protein is by using a small protein that binds to it. That was developed by collaborators of ours in the Netherlands. And when we got it to our lab, a student named Andrew added a fluorescent tag to that little protein. So we have this fluorescently tagged little protein that binds the Huntington protein, and we can basically watch it in live cells and see what the Huntington protein is doing. And you can see it moves to the nucleus, which is that round part in the middle of the cell, and that's where the DNA is stored. So through further experiments, Laura also worked out exactly which bit of the Huntington detects the oxidants and how that causes the Huntington protein to move to the nucleus. So, and it turns out that that little bit is right next to the expanded part that has the extra amino acids. And we know that that little bit it doesn't behave properly when you have those extra amino acids there. So this is basically another piece of the puzzle that Laura's worked out, and that we'll be able to use this information as we go forward, understanding what the Huntington protein does and screening drugs for things that affect it. In the meantime, I was working on a totally different project. So a group from UCLA published a study in which they found that they could improve the symptoms of an HD mouse model by targeting this DNA repair protein called ATM. And again, this was before the GWAS study told us that DNA repair was important. So we didn't make the connection with DNA repair, but we did notice that this DNA repair protein called ATM has a staining pattern that looks a lot like the Huntington protein. So again, these are cells out of the clinic, and this image was taken on a state-of-the-art super resolution microscope. This is the dark room that Bev was talking about that we do all our work in. They might be dark rooms, but they have really awesome microscopes in them. So this is the nucleus of a single cell. The DNA is stained in blue, and the Huntington protein is stained in green. And you can see the DNA repair protein is stained in red, and you can see that the Huntington protein and the DNA repair protein go to the same spots, down to half a micron, which is like 1/100th the width of a hair. I looked it up. So, you know, when we see two proteins getting together in the same spot, especially at high resolution like this, it's a clue that they might both be working on something similar. So we simply asked, is Huntington have a role in DNA repair? And we have the tools to look at whether Huntington can move to sites of DNA damage, because you can damage DNA with UV light. So we can use a laser and draw a stripe of damage across the DNA in the nucleus. And that's exactly what I did in this experiment. I zapped a stripe of damage across the nucleus, gave the cells about 20 minutes to recover and do their thing, and then I stained them. And I stained the DNA again in blue, and the Huntington protein in green. And you can see, especially in the blow-up, that in that 20 minutes, the Huntington protein gathered at the sites of DNA damage. So this is pretty exciting. We started doing a lot of laser stripe experiments after this, and the student Andrew, who developed the fluorescently tagged little protein that binds Huntington, he tried this so that we could look at it in live cells. So in blue is the DNA, and that dark spot, that's where he zapped it with the laser. And you can see in green, the Huntington proteins all gathering at that spot. So through further experiments, we found that the Huntington protein actually interacts with a lot of DNA repair proteins, especially in response to oxidants and oxidative damage. And so this goes hand in hand with what Laura found, because she found that the Huntington protein detects oxidants and then moves to the nucleus. And now we found that once there, it interacts with DNA repair proteins acting as a scaffold at sites of DNA damage. So I didn't have time to tell you about the other, the third project. Ray's going to tell you about the fourth one. But basically, the point I want to make here is that we were working on 4 seemingly unrelated things. Laura D. was asking, what happens to Huntington on oxidative stress? And she found that Huntington responds to oxidants and moves to the nucleus. I was looking for a relationship between Huntington and this DNA repair protein, and I found that it's acting as a scaffold for these DNA repair proteins at sites of DNA damage. Susie was looking at what controls Huntington's location in different places in the cell. And what she found was that the protein that's in charge of deciding whether Huntington is in the nucleus or not is a DNA repair protein. We'll have to tell you about that next time. And Laura B. Was screening drug candidates for things that affect the Huntington protein. And this is really the most exciting thing. The thing that she came up with turned out to be a natural product that our bodies produce. It's a natural product of oxidative DNA damage repair. And Ray's going to talk about this. But the main thing is that of all the things that Huntington does in the cell, and of all the things that we've looked at before of its jobs and what might be going wrong, we think we're on the right track now because the GWAS data told us that DNA repair and oxidation are important. So we're looking at the right thing. Okay, back to it.
Okay, so what can we do about this? So we find out Huntington's involved in DNA repair, and there is a long list of neurological diseases that are defective in DNA repair. The ATM protein, ATM stands for ataxia telangiectasia mutated. It's actually a neurological disease. Okay, so now on to what my lab does best in our technology, right? And it starts with an entire system of unbiased screening for molecules. So we have the ability these days to get libraries and thousands of molecules, and we need a way to sort through them, number one, in a practical manner, but number two, in an unbiased manner, because we have a lot of DNA. So I'm going to demonstrate why humans are no good for looking through microscopes. When you think about a microscope, you think about your high school microscope, your high school biology class. You put something underneath and you fiddle with the focus knob. Modern microscopes, like the ones in my lab, don't even have oculars. You don't even look at them. You let the computer look at them, right? So I'm going to flash up an image, and I want you guys to just immediately register in your mind what you see in that image, okay? Here's the image. Okay, so about half of you in the room saw the shadowed image of a woman's face, right? And about half of you in the room saw a guy playing the saxophone. So you can see the saxophone. But you see what the problem is. This is what's wrong with humans. We're all looking at the same image, but we're calling it two completely different things. And it's no different than when we look at microscopy images, even as highly trained scientists, even as highly trained medical professionals. So there's a couple of famous studies out there about the notorious unreliability of radiologists. These are the guys that look at x-rays in the hospital and will look for stuff within the x-rays and then define whether or not you have a certain disease or not. And people have actually found, and this is published about a year ago, that pigeons can do a better job than humans at identifying you. So if you flash up pictures of cancerous tissue microscope images, if you train the pigeon as to what is cancer and what is not, it'll be able to sort it eventually with 99% accuracy, which is better than having 3 board-certified radiologists staring at that image. Okay, so the future of medicine is pigeons. I'm sorry, Ed. You're about to be sacked. All right, so we're now 100% human-free in our screening, right? So we have liquid handlers, these robots that can take 384 different compounds at once and move it from our storage plates onto our cells of interest. Our cells are no longer model cells. We're looking at cells out of humans from the clinic, right? All of that gets fed into robotic microscopes that will go through and take as many images as we want in each one of those wells and generate a tremendous amount of data. Then guys like this, and you'll note this is a 3D propeller, will then put them together in various ways in computer algorithms to sort the images. So we're feeding a bunch of compounds randomly into HD cells, and we are screening them by computer algorithms. There's no humans involved from front to back. And results of this are plots like this, where we get a lot of compounds in the middle of that big glob that are doing nothing. Really, truly nothing is in the middle of the glob because we didn't do anything to those cells. And as stuff moves further away from that center of the glob, it tells us that our compound is doing something. It doesn't tell us what it's doing, but it says it's doing something to our system, and we should take a good look at it. And one of those compounds that we pulled out of these types of screens is highlighted up there as mutant compound number 50, a big blue arrow. Okay, so we've done a lot of subsequent work with that compound number 50, and we at some point got it into mouse models with our collaborator, Simonetta Scipione at the University of Alberta, who's done a lot of model work in HD. I'm not going to explain to you what's in every one of these figures, but we dosed these animals at different doses by different modalities. We gave it to some of them, we injected them in the belly, some of them we fed it to them in their chow for about a month. So we wait for the mice to get sick, and we score how sick they are, then we give them the treatment for about a month, and then we score the mice again. And what we can see at one dosage is correction of a lot of the problems that we see in the HD phenotypes in these mice. At very low dosage, just in the chow, so just putting this compound into their food, we can see correction of a lot of the phenotypes that appear that are closely related to the psychological phenotypes in HD, so what they call anxiety measures in the mouse. And you can see this is a profound effect. So, but the big question is, in pharmaceutical industry, is what you're seeing because what you think is going wrong, or you think is going on with your compound. So they want to know, do we have target engagement? And what that means is, when you give your compound to your animal model and it has a positive effect, is it doing that because of why you think it's doing that, and not for some other reason? So the easiest way to look at that in Huntington's disease is look at the Huntington protein itself. So we treat the animal with this compound, it gets better for these phenotypes, but it only gets better certain phenotypes, not completely. Then we're going to look at their brains, and that's what Simonetta did with her technician. And this is the kind of raw data that we stare at the labs. These look like little tick marks here. That's the actual way that we quantify the protein in the different brain regions of the mouse. And what we could see is that in this dosage, in the way we gave this compound to the mouse, we could see a dramatic reduction of the mutant Huntington protein in one region of the brain and not another. And where we did reduce the region of the compound in that region of the brain makes sense from what we know that brain region's functions in relation to things like anxiety and the psychological and psychiatric measures in these types of mouse models. Then, if we look at gross things like these aggregates that people have been talking about for years, they're completely gone in certain parts of the brain. So we have a tremendous effect here. And this is a small molecule that we can deliver through food, and it passes through the blood-brain barrier, and it's already been done, passed, and gone through clinical trials for disease. And it's known safe in humans. Now, is this a drug? No, it's not, because it doesn't work very well. The problem is, as the drug is getting in from the outside of the brain towards the inside of the brain, it's getting quickly burned up. And the reason for that is because it's a natural metabolite. And so we need something that works better. So I'm now working with a company called Mitochine, and I have to state a conflict here because I'm actually a shareholder of this company now. But these are a group of experts out of UC California and San Francisco who've generated analogs of these compounds. So versions of this compound that we've already screened, and we can find that they work not just a little bit better, but a lot better, even though we needed a surprisingly small amount in the mouse. So we are developing these analogs. We think that somewhere within these analogs, and there's not a lot of them, because there's only so much we can do with chemistry of this compound, there's something in there that's going to be a potential actual drug. So overall, what does this make us think about Huntington's disease? Well, it could be the reason why this is an age-onset disease. We know that what happens from when we're born, example over there, to older people, there's a steady increase in oxidative stress. And really the brain gets hit hard with this because of all the energy that your body uses at rest sitting right now, over 50% of it occurs above your neck in the brain, right? There's a tremendous metabolism going on within the brain. And we think that the early pre-manifest stage of HD is actually when there's this defect in DNA repair that is leading to that CAG expansion that Jeff talked about, right? And then finally you hit a threshold, and then you get disease from what we see in the clinic. Now, we've been focusing on the gray box, on Huntington's disease, and when the point of time when people get sick. But this suggests that maybe we really need to look at the pre-manifest stage a lot harder in terms of paradigms and how we're going to treat people in the future. Nevertheless, these compounds that we are developing now do work, apparently in the mouse model, to take us from a disease state back to a normal state. And it's quite striking what effect it is. So, the concept is that DNA rusts as we get older. Here's a great Neil Young album, Rust Never Sleeps. And the idea is here, the analogy is a rusty car. If you have a car and your car starts to develop a little bit of rust around the corners, you can leave it, you're fine. You can drive for years with that rusty car. If you don't care about it, like who cares? Just keep going. But eventually, it's going to catch up with you. And the rust is going to accumulate, and eventually your car is not actually going to work, and you're going to get broken down on the side of the road. It's going to crumble into dust. So, there is a problem of DNA repair in Huntington's disease. There are ways with small molecules that apparently we are fixing this problem. So now we have an interesting new concept. In addition to the concepts of gene silencing, turn off the mutant protein, there may be actually ways to fix the mutant protein so it now works like the normal protein, which means that we're not losing any functions of this protein, and it could be beneficial. All right, so the first question that I know is going through all your minds is, what's the time frame? Every time I talk to you guys, I always give you this, well, in 5 years, it's going to be whatever. So, enough, when, right? This is what you want to know, when. So, here are the list of things that need to be done, and this is a classic list of things that need to be done in drug discovery. We need to make analog compounds. We need to test the compounds in our various models in our mice. We need to do what are called pharmacokinetic and pharmacodynamic studies. So we need to understand how these compounds are metabolized, how long they last, how quickly they're cleared, how effective they are, how safe they are. We need to patent it all. Now this might seem like greedy capitalism, but the truth is, if we don't have a patent on a molecule, no pharmaceutical company is going to carry us through clinical trials. And that's where the big expense is in drug discovery. We need a pharma partner. Right? So we need a biotechnology partner to help us develop the compounds, and we need a pharma partner to help us carry through the clinical trials. And to date, everything in green has been done. So we're close, right? We are just dosing analog compounds now into mice, and we are working very hard with our biotechnology partner to talk to large pharmaceutical companies about all of this science and all of the preclinical mouse data to say it's time to think about generating a clinical trial. And what does this say about the future of Huntington's disease? And thinking about it in treatment. And I will give you an animal. So this compound is a normal human metabolite, right? It normally exists, but it's absent in Huntington's disease. That could define Huntington as a metabolic disorder, which is very similar to a disease called phenylketonuria, PKU. So some of you might know what PKU is. Every baby born after sometime in the 70s, including that one, was tested for PKU at birth. That's what they do when they take that little blood sample with the heel prick when the baby's born. They're testing for PKU because they know that if you don't diagnose PKU from birth and you feed that child a normal diet, it will develop severe mental retardation and severe developmental problems within the brain and really bad neurological disorder. But if you go and look up these PKU organizations, there's all kinds of testimonials of kids that were tested 20 years ago who were put on special diets, who then went on to just be completely normal. It's a difficult life on these diets. It's not easy, but they don't develop the disease. And this may be the future of how we think about Huntington's disease. Okay, so I'd just like to thank all of these people. There's a lot of people involved in this. There's a lot of labs involved in this, and this is growing exponentially. I first presented this data in Palm Springs in last February, and I asked the community to go out and take this compound and try it in their assays, and a lot of them have done it, and a lot of them have reported back positive results. So there's a certain level of excitement on this, and hopefully 2 years from now, we're going to be talking about very exciting stuff. So I'll be glad to take any questions. Get Tamara back up here to answer any questions as well.
So along with that, what we said there's a repair, but again, repair cells that aren't there. So, I'll let you stop where I'm at.
Okay, so the question is, right, so you can't repair cells that aren't there anymore. Presumably what we can do for those affected is slow or halt the progression of the disease. But it's impossible to know until we go to clinical trials. One of the parts of this story is, years ago, I came to you guys and I said it was time for the research community to get out of our labs and start using HD patient cells. And you guys gave us everything we needed. There's repositories at Correlated that I have access to. Call them up. At minimal cost, I get these things delivered in about 3 days. This is really about the feedback scientifically from the patient community. So the question that we don't know is how much benefit will we get if we can stop the disease progress in HD, because the brain is a very interesting organ. It's plastic. It has the ability to repair itself. If the disease is still always occurring, you're not going to be able to see that repair. But as you know from people who've had massive strokes, entire parts of the brain can die off. But if you give that person the right therapy, they can rewire those connections within the brain, and that's called synaptic plasticity, and the human brain does this. So the brain will self-repair if we can stop the disease. But we don't, this is why we need trials, right?
The youth tested into the system, so you see a better picture for them.
Potentially. Yeah, potentially this could mean, a different way for youth to think about whether this situation, whether they're at risk or whether they want to get tested or not. Yeah. But we need to get good science first. Yeah.
So you talked about this being a normal metabolite in the body. So would this be something that you imagine it would be like would you would have a diet thing? Is that would it be lifestyle things to change as you're growing up, or more something to be given to increase that amount of metabolite?
So the problem with giving, so the question is, he's asking, this is a normal human metabolite. So can we make a much closer analogy to phenylketonuria, where we just change the diet, for example, to get this metabolite back in? And the answer is no, because what it means is this is a metabolic product of DNA repair, right? And DNA repair isn't occurring in HD. You put it back and it looks like we can reset a cascade of things that are going wrong. But the problem with it is the reason why we don't go to clinical trial with this molecule is because it's supposed to be excreted from the brain. You actually find it in human urine because we're supposed to be getting rid of this. This is kind of our trash, right? But as trash, it also has important signaling roles that tells molecular mechanisms that there's something wrong with the DNA and you need to do this and you need to do that and you need to do the other thing. So that's why we need the analog compounds. We need things that will trick the body into getting all the same signals but not want to clear this thing out of the way, and which will be beneficial, presumably, in an HD situation. Just yell, Susan, you're loud.
Okay, is this on?
Yeah.
Yes, okay. You were very clear that you're very close. Like, can you hazard a guess as a date this might go to clinical trials?
No, we don't know until, it depends on the science in the next, it depends on the science, but in the science in the next 12 to 18 months. We're funded to do this. I've got good amount of support through the HSC from the Krimble Foundation in Toronto, enough money for us to answer those questions, and then we'll have some real timelines on where to go forward. And I think that in that period of time, we're essentially going to accumulate the evidence that would be able to tell the pharmaceutical company this is worth investing in, because we really need their help. The amount of money needed to do a proper clinical trial is just beyond the Canadian government or organizations like this. We need pharma at some point. It's just a reality. It's hundreds of millions. Any questions for Tam?
When you were talking about the study that looked at the variability in age of onset, and it came down to like those three things, and oxidative damage and DNA damage, was there anything, like other than genetics, anything environmental that they found?
So I actually don't know, did they,
No, they were only
control? So they basically, they looked at the DNA of these patients, and it's my understanding, maybe you guys know, that they had the DNA there. So they didn't have to bring in 4,000 people, right? So they didn't actually have information necessarily on those 4,000 people and what their lifestyle was like. So strictly genes.
So the information that they had, because it was a huge number of people, the information that they had about what those people had done during their lives was very, very little. So basically, the only thing that they had for everyone was the age of onset of the Huntington's disease. So they had the whole genome and then age of onset data, and that was it. There have been studies of environmental factors that influence onset. We haven't really found any that reliably predict or influence onset, so that we don't really have good advice to say, eat blueberries or go, use, you watch this TV channel or whatever. What the best evidence that we have is that being active rather than not active has provided the strongest signal to date about some environmental or lifestyle thing that is likely to be beneficial in HD. But we haven't had any kind of trial or interventional study that's been able to look at that.
So exercise is good for you.
Well, we knew that already.
Yeah.
Probably even more good in HD.
It's actually true in all neurodegenerative diseases. The benefits of exercise are pretty significant.
Any other questions?
I think our job is done here. Thank you.
Thank you.
HD is really complicated. Aren't we lucky we have really smart people working on it?